This video is about Chapter 3, Cell Division. Subtopic, Mitosis. So after the lesson, you should be able to describe the 4 stages of mitosis, the behavior of the chromosome at each stage, and describe briefly the process of cytokinesis in animal and plant cells, and compare cell division in animal and plants. So mitosis process is where the cell growth stops after the process of cell cycle and ready for the cell division at this stage. Cellular energy is focused on the orderly division into 2 daughter cells. So mitosis consists of 4 phases, which is prophase, metaphase, anaphase, and telophase. If according to the latest Campbell 11 edition, mitosis has 5 stages, which has the extra pro-metaphase here. Okay, but according to our syllabus here, we're using these 4 stages. After the 4 stages, it follows by the division of cytoplasm, which is called cytokinesis. So definition of mitosis is the division of the genetic material in the nucleus, followed by process of cytokinesis. So the result of mitosis, it will produce 2 daughter cells which having the same number of chromosomes as the parent cells. So mitosis has the 4 stages: prophase, metaphase, anaphase, telophase, while cytokinesis can be divided into animal cell and the plant cell, where animal cell undergo cleavage furrow, plant cell has formation of cell plate. So now let's see the detail of prophase. So this is the diagram of prophase under the microscope. Okay, so during prophase, the nuclear envelope and the nucleolus will disintegrate. Disintegrate, in other words, to say it disappear, which means it is not visible under microscope. So chromatin at this moment, they become shortened and thickened. That's why under the microscope, we are able to see the chromatin better, and at this stage, we call it as a chromosome. Okay, so it is visible under the light microscope. It's more tightly coiled, more condensed, okay, into a discrete or compact chromosome. So each duplicated chromosome appear as 2 identical sister chromatids which join at the centromere. Okay, something like this. So the centromere in the center, and they have a visible chromosome and 2 sister chromatids. So the mitotic spindle or the microtubules begins to form. So during this stage, the ester that extend from the centrosomes also form. Okay, so ester is like a star-shaped structure, starts to form during prophase, and the centrosome move away from each other, apparently propelled by the lengthening of microtubules between them. So the centrosome move away from each other, move to the opposite pole. And the description, the meaning of the centrosome here. Okay, the main center for the cell that organizes the microtubule and responsible for organizing the spindle fiber. Okay, so this is the process of the prophase, where we can see the centromere holding the sister chromatid. Okay, in the form of this centromere, in the form of sister chromatid. So why we call it sister? Because both of these come from the same DNA content. This come from after the process of DNA replication. So both of these have the same DNA content. Okay, come from the duplication of the chromatin or the replication of DNA. So the nucleolus, okay, and the nuclear envelope starts to disintegrate, and the centrosome move to the opposite pole, and early mitotic spindle starts to form. Okay, and the ester begins to form. Right. Next is the metaphase. So metaphase is the longest phase in the mitosis. Okay, so the centrosome are now at the opposite pole of the cell. A chromosome as a sister chromatid move and align on the metaphase plate. Okay, metaphase plate is the middle line of the cell, which we also call it as equatorial plate. So chromosome centromere lie on the metaphase plate. For each chromosome, the kinetochore of the sister chromatid attached to the kinetochore microtubule coming from opposite pole. Okay, so kinetochore microtubule. Now for this sister chromatid, they have some protein-like structure. We call it kinetochore here. So kinetochore microtubule is the microtubule that attached to the kinetochore here. Okay, it besides the centromere. So this is the metaphase plate in the middle of the cell. Okay, so now this centromere they align on the center of the metaphase plate. Alright, so this kinetochore microtubule, they just attach to the kinetochore. Okay, and our centrosome with the centriole has already reached the opposite pole. Okay, next phase is anaphase. Okay, so anaphase is the shortest stage of mitosis. Now centromere will be split, allowing the sister chromatid to separate. So our sister chromatid will be separated. Shortening of the spindle fiber or shortening of the kinetochore microtubule. Okay, because of the kinetochore microtubule, they start to become shortened. So when they shorten, they will pull away the sister chromatid, which cause the sister chromatid to be separated. Okay, two sister chromatid separated. So now we call it as a daughter chromosome. Now we no longer call them as sister chromatid because they are no longer holding at the same centromere. So now the daughter chromosome is moving towards the opposite pole. So daughter chromosome will move the centromere first because the microtubule kinetochore are attached at the centromere region. Okay, means when the microtubule shorten, it will pull our centromere first. That's why we do not draw it like this when they separated. Okay, this is wrong. Because when it pull the centromere first, they will move like this. Okay, in the opposite direction. Okay, so this is the correct one. So this is the wrong one. So now the cell elongate as the non-kinetochore or the polar microtubule will lengthen. So for the microtubule which do not attach to the chromosome, you call it as a non-kinetochore microtubule. So this non-kinetochore microtubule, they will lengthen and cause the cell to elongate. So when the cell elongate, which means it is preparing for the cytokinesis process. So non-kinetochore microtubule from the opposite pole overlap and push against each other and elongating the cell. Okay, so the mitochondria here will provide energy for the movement of chromosome, And by the end, two ends of the cell have equivalent and complete the collections of chromosomes. Okay, so now it's almost completing the mitosis process where each cell has the same chromosome number as the parent cell. And the last phase is called telophase. Okay, so in telophase, chromosome reach the opposite pole of the cell. Okay, so they already reach the opposite pole. Two daughter nucleolus are reformed. Okay, the new nuclear nuclei form around each daughter cell. Spindle fiber will disintegrate. Okay, chromosome become less condensed. Okay, less condensed as beginning of the process, which are uncoiled and lengthened, thus become invisible again. Okay. Okay, so mitosis, the division of one nucleus into two genetically identical nucleus, is now complete, and the cytokinesis begins at late telophase. Okay. So now the telophase and the cytokinesis, when the process of the telophase almost towards the end, which is the late telophase, cytokinesis begin to occur, where the process of cleavage furrow starts to form in animal cell. Okay, so under microscope, we should able to identify for this cell, they are undergoing prophase. Okay, prophase, because we can see the chromosome clearer. Okay, and this one is the metaphase, where the chromosome align on the metaphase plate. Anaphase, when it is separated to opposite pole, while telophase, they already reach to the opposite pole. Okay, so this will be the prophase also. Chromosome become visible. Okay, so comparison, what's the difference of anaphase and telophase? So from the diagram here, we can see the anaphase, the chromosome is still quite visible and they just started to move to opposite pole, while for telophase, it's almost in the opposite pole already. Okay, so we can see that kinetochore microtubule shorten, separating chromosome to opposite pole. Polar microtubule elongate, preparing the cell for cytokinesis. So for telophase, the chromosome already reached the opposite pole, where kinetochore already disappear. Polar microtubule continue to elongate, preparing cell for cytokinesis, and the nuclear envelope starts to reform. Okay, nuclear envelope starts to form, nucleolus starts to appear again, chromosome decondense. Okay, cytokinesis process is the division of the cytoplasm to form two separate daughter cells. And division of the cytoplasm occur by late telophase, so the identical daughter cell will appear shortly after the process of mitosis. So we can divide cytokinesis into either in animal cell or in the plant cell. So we look at the cytokinesis in animal cell first. So the process is known as the cleavage process. So for cleavage process, the first sign is the formation of a cleavage furrow. So furrow is like a gap, okay, between two areas. So a shallow groove in the cell surface near the old metaphase plate. So the cleavage will occur through the furrowing, okay, where the membrane is pulled inward. So the membrane will be pulled inward, okay, by the cytoskeleton, the skeleton which makes the cytoplasm. the shallow groove is formed in the cell surface near the O metaphase plate, and a contractile ring of actin microfilament formed on the cytoplasmic side of the furrow. So eventually they will be pulled and shortening until the two cells separated. Okay, so these actin microfilaments will interact with the protein myosin, causing the ring to contract and reducing the diameter. Okay, until they deepen and the parent cell pinch into two cells. Okay, after they pinch into two cells, this will be separated, split off, and they form two completely cells, which each has a complete nucleus. While cytokinesis in the plant cell is forming the cell plate. Okay, the cell plate, because the cell wall is not able to separate it. Okay, so they form the cell plate in the center here instead of cleavage furrow because of the cell wall. Okay, so cytokinesis is in the plant cell. First, they form a line of vesicle. This vesicle is originate from the Golgi body. Okay, originate from the Golgi body inside the cell. So the Golgi body form the vesicle. So now this vesicle will construct both the primary cell wall and the middle lamella. Okay, so this vesicle will be enlarged and fused. So they become larger and larger. Eventually they fuse together to form the cell plate, form across the equatorial of the O cell. So they form two membranes which grow laterally or expand outward and fuse with the existing membrane. So after that, they form two new plant cells, each with its own plasma membrane. So a new cell wall has formed between the membrane of the cell plate. Okay, it arises from the content of the cell plate. Okay, so this is the vesicle, and after that, all the vesicles join together and they become a new cell wall here. Okay, so this is the formation of cleavage furrow and the cell plate in the plant cell. So now we compare mitosis in the plant cell and animal cell. So similarity for both cells, both involve four phases, which is prophase, metaphase, anaphase, and telophase. Both occurs in the somatic cell. Somatic cell is the cell other than the sex cell, which is the diploid cell. Okay, diploid cell. Both also produce two identical daughter cells, means 2n parent cell produce 2n daughter cells. Okay, while the differences between the mitosis in plant and animal cells, for animal cell we have the centriole, okay, present in the region called centrosome, while plant cell they do not have centriole, they only have the centrosome. So due to the centriole, they form the ester. So for plant cell, they do not form ester. So cytokinesis in the animal cell is forming the cleavage furrow, where it forms the shallow groove in the cell surface. Cleavage is growth from outside to inside, okay, outside to inside. While for plant cell, cytokinesis is forming the cell plate. Okay, it forms the new cell membrane that separates the cell into two daughter cells. Cell plate grow from inside to outside. Okay, so let's say this is the plant cell. So the vesicle is formed from inside and then they join together. So inside to outside to form the new cell wall that separates the cell into two daughter cells. So animal cells occurs in all the somatic cell, while plant cell occurs mainly in the meristem tissue, which is root tip and shoot apex. Remember, we say that the meristem tissue is a young and immature cell. They keep continuing to undergo cell division. Other cells such as the permanent tissue, parenchyma, collenchyma, sclerenchyma, they do not undergo cell division. Okay, so what is the importance of mitosis? To provide genetic stability to the cell, which maintain the genetic content of the cell. To make sure that all the daughter cells have the same chromosome number as the parent cells. So daughter cells are genetically identical to the parent cell. There is no variation. Means all the daughter cells are exactly the same like the parent cell. So there is no variation. So this will make sure the genetic stability within the population of the cells derived from the same parental cell. So if there is any difference which occur in the daughter cell, then you call that mutation has occurred. Okay. Second importance is for the growth and development of the cell, where the number of cells will increase. So this is the basis of growth in the multicellular organism. Example, growth or increase in the body weight or organ development. So from one cell, they grow into 2 cells, 3 cells, 4 cells, 5 cells. So it will grow the organism. The third importance is for the cell replacement or regeneration. When any cell is dead, aged or damaged, it will be replaced by the new cell. Number 4 is for regeneration, where some of the animals are able to regenerate the whole parts of the body. Example is like the legs in the crustacean, okay, arms in the starfish. Production of the new cells involve the mitosis. Okay, asexual reproduction. Mitosis is the basis for asexual reproduction. So for those organisms who do not undergo sexual reproduction, so asexual reproduction will be the process which they use to produce the new identical individual of a species by one parent organism. Okay, example is vegetative reproduction, which occurs in the plants.